Hello everyone, we are BIOCLINIC, a firm focused on providing you the world's best in genetic testing. Today we are unveiling a new test for Parkinson's disease. Parkinson's disease is a debilitating illness that affects about 13 per 100,000 people in the United States and about 4 million people worldwide. The most common symptom of the disease is the shaking and trembling of one's limbs. Additionally, the disease can affect emotions and cognitive abilities. This disease is caused by the impairment or death of nerve cells in the substantia nigra and is presumed to be based on genetic predisposition. Our competitor, 23andMe, tests for some parts of this disease. To discuss this farther is Annaya.
23andMe does genetic testing for Parkinson's disease using the LRRK2 and GBA gene. More specifically, 23andMe will test for the G2019S variant located on the LRRK2 gene. This gene is present in neural tissues that are located in our brain and throughout our body. The LRRK2 gene with the G2019S variant expresses an increased risk of developing Parkinson's disease. Here's how. The LRRK2 gene produces a protein called DARDane. DARDane produces an enzyme function known as kinase activity that helps to transmit signals between the brain and other cells while assisting with the transfer of phosphate groups that help to turn the cells off and on. Testing for the G2019S variant is most common in familial Parkinson's disease.
For our purposes, BIOCLINIC strives to provide a superior test by testing for more genes than 23andMe. Specifically, we wanted to test for the PRKN gene, a gene that is highly correlated with Parkinson's disease. Our SNP chip test will test for the PRKN gene. To discuss the details of this gene, here's Jackson.
The PRKN gene is a gene that encodes for the protein parkin. Parkin plays a critical role in the regulation of protein degradation and mitochondrial function, which are essential for maintaining the health and function of neurons in the nervous system. Some mutations in the PRKN gene lead to the production of abnormally small parkin genes that are non-functional and rapidly broken down within normal cells. This can lead to the accumulation of toxic proteins and oxidative stress, which can damage neurons and contribute to the development of Parkinson's disease. Parkin is primarily expressed in cells of the nervous system, including neurons and glial cells, as well as other cell types such as muscle cells, heart cells, and pancreatic beta cells. However, the expression levels of Parkin can vary depending on the cell type and the physiological condition. Variations that change the structure or expression of the Parkin protein can contribute to the development of Parkinson's disease. Mutations in the PRKN gene itself can disrupt Parkin function, and variants in other genes that regulate or interact with Parkin could also affect its function and contribute to the disease. For example, mutations in the PINK1 gene, which encodes another protein involved in mitochondrial function, can disrupt the interaction between PINK1 and Parkin, leading to impaired mitochondrial function and the accumulation of toxic proteins. In summary, the PRKN gene and its protein product, Parkin, play critical roles in maintaining the health and function of neurons in the nervous system. Variations that change the structure or expression of Parkin can contribute to the development of Parkinson's disease, making the PRKN gene an important genetic variation to consider when testing for genetic risk for this trait.
Thanks, Jackson. To further discuss some limitations and ethical considerations, here's Ella.
Parkinson's is a complex trait, so there's still much unknown about its causes. As of right now, there are 7 known SNPs associated with this inheritance. However, the 23andMe test only accounts for changes in the LRRK2 and GBA genes. Our test would be more comprehensive because it would account for changes in all 7 possible SNPs, making it be able to test more genetic variability. Parkinson's does not follow traditional inheritance patterns, so as far as ethical considerations, this test is not perfectly predictive. It may not be in someone's best interest financially and emotionally, especially if they don't really have familial history of Parkinson's. This test would need to use a SNP chip test, which is more expensive than traditional gel electrophoresis. However, if a person does have familial history, this few $100s might be cheaper than the medical bill racked up with later detection of the disease. Also, if you're someone who has hypochondriac tendencies or gets overly anxious about these kind of diagnoses, it might not be the best for you, especially if you don't have familial history of the disease.
Finally, here is Annaya with some concluding limitations.
But it does have its limitations. One is that the physiological function of the LRRK2 gene is largely unknown, making it extremely difficult to pinpoint an exact cause for the Parkinson's disease onset. Furthermore, the LRRK2 gene mutation is only common in about 2 to 7% of familial cases, and it is inherited in an autosomal dominant pattern, meaning that if you have any family history with Parkinson's disease and its mutation, you could already be aware of your high risk at developing Parkinson's. Moreover, choosing this test may only be beneficial to those who have familial Parkinson's disease. If you fall in the other 90% who are unsure at their risk of developing Parkinson's disease, you should consider our BIOCLINIC genetic testing for the PRKN gene and the risk of developing Parkinson's.
As you can see, Parkinson's disease is a complex trait that requires intense genetic testing. But BIOCLINIC is up to the job. Here are our references. And I could not think of a snazzy way to do this, so this is the best you're gonna get. We hope to test your DNA soon with BIOCLINIC. BIOCLINIC.